Okay, hi folks. So this is a podcast version of the Molecular Biology Techniques lecture from the Molecular Genetics module, Year 2 Biomed. What we're going to be talking about for the next 45 minutes or so are some of the technical details that underpin, the major core methodologies that you would use were you to pursue a career in modern molecular biology. Specifically PCR and derivatives of PCR technology, and some of the hybridization techniques which kind of go hand in hand with PCR for molecular biology research and for diagnostic work as well. So those of you who remember the module at level 1, human genetics, will have seen this slightly maniacal looking character here before. This is Professor Kary Mullis, a Nobel Prize winner. He's a guy who invented PCR in the 80s. And we're going to start the talk today by revisiting PCR in a little bit more detail than we covered it last year, and also looking at some more advanced variations on that technique. So specifically we'll start with a bit of a history of PCR and the underlying technology of basic or conventional PCR itself, some of the applications we can use for PCR. We're also going to discuss relatively recent, although not, I guess it came around in the early part of the last decade, so just in the start of the new millennia. An advancement in the PCR technology known as real-time PCR, and we'll talk about what real-time PCR is, how it differs from conventional PCR, what advantages it offers us as research scientists or diagnostic scientists, and what some of the applications of that advanced technology are. What we'll do then is we'll go on to discuss some of the classical hybridization technologies which exist in molecular biology, specifically southern blotting, western blotting and northern blotting. Each of these different techniques is concerned with the analysis of a different product from the genome. Specifically, northern blotting is particularly interested in RNA. Southern blotting is interested in DNA, and western blotting is interested in protein. And we'll discuss how each of these different techniques works, how we actually carry out the techniques, and what they're actually used for with regards to detecting these different things on gels and membranes, etc. And we'll finish with a summary, just to pull everything together. So really what you need to take away from this lecture is, firstly, you need to understand the technology that drives PCR and real-time PCR. Particularly, you need to understand what components go into a PCR to make it work, what those components actually do in that PCR reaction, and how you can manipulate those components to try and troubleshoot a PCR reaction that isn't working. You also need to have an appreciation of what real-time PCR is, the chemistry that underpins it, and how it can be used to do more advanced or carry out more advanced analyses of gene expression, etc. And secondly, you need to be aware of the common hybridization techniques and understand how they differ. One or two of these techniques have been replaced now by real-time quantitative PCR. However, western blotting is still very used at a very high level throughout research and in diagnostic situations also. So it's important you have an understanding of all of these techniques by the end of the lecture, and by the time you've finished engaging with wider reading, of course. So here he is. We've seen him many times before, Kary Mullis. Perhaps it needed somebody of Kary Mullis's special intellect, stroke, class A drug-induced haze to actually come up with the idea of PCR in the first place. It was a bit of a conceptual leap forward, really. What Mullis realised, that is that you could, in essence, in a tube, using a few simple reagents, copy what goes on in the cell during DNA replication. So as you'll find out next year in medical genetics, DNA replication is a very complexly regulated event during cell division. However, the actual nuts and bolts of how you make more DNA from a DNA template is actually quite straightforward. You don't need that many things to make that actual chemical reaction work. And that's, I guess, what Mullis realised, and he also realised that one of the major obstacles to actually producing new DNA from existing DNA was circumventable via the use of enzymes which were isolated from special type of bacteria which were heat resistant. So we'll talk in more detail about what exactly these elements are now. So firstly though, just to start with a quote from Mullis, the beauty of PCR really is that beginning with a single molecule of the genetic material, DNA, PCR can generate 100 billion similar molecules in an afternoon. The reaction is easy to execute, requires no more than a test tube, a few simple reagents and a source of heat. So, you know, it's simplicity that basically makes PCR such a fantastic technique, such a fantastic method to have at your disposal when you're carrying out research in the lab. It's cheap, it's safe, it's quick, it's robust, it works 99% of the time as long as everything's set up correctly. It's a really, really robust, powerful technique. And the underlying mechanism of PCR is essentially based on a basic understanding of nucleic acid biochemistry, the sort of thing you'll have covered in biological molecules, I'm sure, and cell biology, and partially in human genetics at level 1. Certainly you'll have encountered it at A level if you did A level biology. And that, in essence, is that in order to make more DNA, you essentially need just a few different things. You need a DNA template, you need some short oligonucleotide DNA primers. The reason you need those primers is that DNA polymerase, the enzyme which copies new DNA, cannot just bind to a template and start making DNA. It requires a short primer region which generates a double-stranded portion to the template in order to bind and then start making new copies. And obviously when it makes the new DNA, it needs to make the new DNA out of something, and that something is these deoxynucleotides. So these individual A's, T's, C's and G's which go to make up the new DNA strand. So Mullis realised if you threw these 4 things into a test tube and applied heat in the right order and at the right amount, you could in theory make more DNA. The trick really was this enzyme here, TaqDNA polymerase, which is the DNA polymerase that Kary Mullis realised would enable him to do PCR as a chain reaction, as an exponential event in an afternoon, as opposed to a rather long-winded and inefficient method which had been used up to that point. So obviously one of the major issues about PCRs, we'll see when we look at the actual steps required to get it to work, is the fact that at some point you need to heat the PCR reaction up to a point which causes the disruption, the melting of the double-stranded DNA that you're using as a template. Now to melt double-stranded DNA, you need to heat it to quite a high temperature, 92, 95 degrees. That causes the DNA double strand to melt. The hydrogen bonds that are effectively holding together the base pairs dissolve in effect, no longer cease to function, and the 2 strands separate. Once the 2 strands separate, you then have the basis for complementary base pairing between the template strands and the primers, and that's then what allows a PCR to go ahead. However, obviously once you've heated a reaction up to 92 degrees, any enzymes in there, which normally operate at 37 degrees from bacteria, tend to be pretty much screwed, tend to be completely denatured and certainly vastly reduced in efficiency and activity. However, Mullis realised that if you were to use actually a DNA polymerase enzyme which you obtain from one of these bacteria that lived in these hot springs, such as a bacteria Thermus aquaticus, then the enzymes of this bacteria must be very resistant to heat. This is a bacteria that exists in hot springs upward of around 80 degrees Celsius, and rightly, as Mullis suspected, it turned out that this was exactly the case, that this DNA polymerase from Thermus aquaticus, or TAC DNA polymerase, was very heat stable. You could heat it over and over in repeated cycles at 90 degrees without very much reduction in activity at all. So that was the final piece of the jigsaw of PCR really, this thermostable DNA polymerase. So how does PCR work? Well, there's a link to a PCR animation here. It's very similar to the one I showed you guys in human genetics, and if you click the link it should take you to the web page. But I don't want to go over that now in this podcast. I don't think it's necessary. You can do that yourself in your own time, just from the normal slides that are available on Blackboard. But I will reiterate what the 3 basic steps of a PCR are. So we've already mentioned the first step, which is denaturation of the template. So generally speaking, the PCR will use a DNA template of double-stranded DNA. So just DNA that might be from a flake of skin at a forensic scene, from a blood sample which has been taken in a hospital, from mummified DNA found in ancient Egypt. Genomic DNA is double-stranded in nature. So DNA exists in its native form as a double-stranded molecule. Now, in order to make PCR work, you need to separate those double strands. In the cell, during DNA replication, this occurs at the replication fork and requires a lot of complex enzymatic reactions involving helicase enzymes which unwind the DNA, etc. It's a very ordered, complex thing which can't be really replicated in the test tube. However, you can achieve the same thing. You can separate the two strands of DNA simply by heating them up to around 94 degrees Celsius. At this temperature, the two strands of the DNA, the DNA double helix, will fall away from one another, leaving you with two single-stranded pieces of DNA. Once you've melted the double-stranded DNA, you then lower the temperature of the PCR to such a point that the PCR primers will anneal to their complementary sequences. Yeah. So remember, a key thing about PCR is it allows you to copy a specific region of DNA. It doesn't randomly copy DNA, it copies whatever region you tell it to copy. And you tell it to copy a specific region by virtue of the design of your oligonucleotide primers. Yeah. So if you do some mathematics on the likelihood of discovering or a genome such as our own, which is around 3-4 billion base pairs, if you do some mathematics on the likelihood of a run of 20 nucleotides, 20 A's, C's, G's and T's, of occurring more than once by chance in that genome, you'll find that the probability is around, well, just below 1. So in essence, what that means is if you design an oligonucleotide primer that's 20 nucleotides long, so for example A, C, G, A, C, T, T, random A's, C's, T's and G's until you have a primer that's 20 bases long, 20 base pairs long, that primer by chance should only anneal to one thing in a genome 3 billion base pairs long. Yeah? So it's just probability. So in other words, a primer that you design will be specific for a specific area, a specific region of that genome. So once these primers have annealed to the to the melted single-stranded DNA template, you then raise the temperature of the reaction to 72 degrees Celsius. The reason you raise it to that temperature is that's the actual optimum temperature for this DNA polymerase from Taq, Thermus aquaticus, to function at. So if you raise it to 72 degrees Celsius, this Taq polymerase starts copying the DNA from the complementary DNA strand to produce a new strand of DNA. And the crux of the matter is that PCR will turn one DNA sequence into 1 billion DNA sequences in just 30 cycles via exponential expansion of the number of sequences that have been copied. Again, that's covered in this PCR animation. It's very clear to see how that works. So what I want to focus on really today and in this podcast is the reagents that go into the PCR. So by now, you should all know what PCR is. You've encountered it in human genetics. You should have a basic understanding that PCR is a technique by which you can copy specific sequences of DNA. If you don't understand that, you need to revisit some of your Year 1 notes and make sure you're familiar with that before you try and understand this next bit on PCR. So what we're talking about now are the actual reagents that you would put into a tube if you were to go to the G-Floor labs now and try and set up a PCR reaction. Yeah, so we've just finished with level 3 projects, third-year projects. A lot of project students have been doing PCR, some unsuccessfully, some very successfully over the last 4 weeks. These are the reagents that those students will have been using in the lab in order to get their PCRs to work. So as you can see, there's a reaction buffer. So all enzymic driven reactions that you set up in a lab will have a reaction buffer. There's also magnesium chloride, which provides magnesium ions to the reaction. We have a DNA template, we have our primers, we have the DNA polymerase, we have the deoxynucleotides, and then we have water. So what I want to do now is just go through what each of these actually contributes to the PCR reaction, so you have a better understanding of what the reagents are used for in the actual experiment itself. So if we start with the reaction buffer, the main purpose of the reaction buffer is to stabilise the DNA polymerase enzyme. So remember, these enzymes are all isolated from E. coli or from other bacteria and operate within the cytoplasm of those particular organisms. Now, the cytoplasm of those organisms contains specific environments with regards to salts, etc., other such things, electrolytes, And what the reaction buffer does is it tries to mimic the natural environment of the enzyme with regards to those salts, concentrations, etc. So that when you run the reaction in a test tube, the enzyme feels at home. It's going to have as much activity, hopefully, in that test tube because of the provision of the correct balance of salts from the reaction buffer as it would have if it was functioning in the actual cell itself. So that's what the reaction buffer does. It stabilises the DNA polymerase. In reality, you get these reaction buffers from companies. They're supplied as a 10 times stock, and generally consist of just a couple of different salts, potassium chloride and tris HCl. Tris is a pH buffer, so it maintains the pH of the reaction. The next reagent is magnesium chloride. It's really there to provide magnesium ions to the reaction, and this is very important because magnesium is an essential cofactor of the DNA polymerase enzyme. So in other words, if you don't have any magnesium ions in your PCR, it's going to severely impair the function of the DNA polymerase enzyme. So activity of the polymerase is going to be drastically reduced. However, and this relates back to troubleshooting PCRs, you need to be aware that having too much magnesium chloride present in a PCR reaction can actually be a bad thing, and result in non-specific priming during the PCR. So generally it's included in a reaction at a final concentration of between 1 and 3 millimolar, the magnesium chloride. So too little, the enzyme, DNA polymerase enzyme won't work properly. Too much, it can lead to non-specific priming. So the primers, rather than just binding to their complementary sequence, can start binding to sequences with 80-90% complementary identity. Yeah. So maybe of those 20 base pairs in your primer, 18 are the same, 2 aren't. Normally that wouldn't bind properly during the PCR. If there's too much magnesium in there, then the PCR becomes a lot less fussy and the primers start binding all over the place. And rather than just producing lots and lots and lots of a specific region of the genome, you produce lots and lots and lots of all sorts of different regions of the genome, yeah? Which is not much use to you at the end of the day with regards to the experiment you're trying to do. So the template, so this is obviously the actual DNA which we're going to copy from. So it must contain the region to be amplified. You can't say that I'm going to PCR up a particular gene and then isolate DNA from a source where that gene's not present. So from a mutant where the gene's deleted, for example. You actually need very, very, very small amounts of DNA template for the PCR to work. In fact, it's often preferable to have too little almost template than too much. And that's for the simple reason that a lot of DNA templates, such as genomic DNA or material that's been isolated from forensic scenes or whatever, contains contaminants. And if you have a high amount of DNA template present in the reaction, you also, by virtue, have a high amount of contaminants. And these contaminants can interfere with how the DNA polymerase works and the efficiency and how robust the PCR reaction itself is. So it's better actually to have less DNA template there. In theory, you need one molecule to get it to work, because by having less there, you've also got less contaminating factors, which can inhibit how well the PCR actually works. So the next thing is very important, oligonucleotide primers. So these primers should be 100% specific for the region you're interested in amplifying. You need a forward and a reverse primer. Again, if you look at the cartoon for PCR, you'll see how this works. Effectively, one PCR primer causes DNA polymerase to copy the DNA sequence in one direction, and then the reverse primer causes it to copy the DNA sequence in the other direction. However, you need to realise that in both cases, DNA polymerase copies in a 5' to 3' direction only. You can only replicate DNA 5' to 3'. It's impossible to do it the other way around because of the chemistry of the reaction. If you've not covered that already, then we will be covering it next year in medical genetics. So because of that, you need to realise that whilst the 5' primer design is very straightforward, it's effectively the same as what the DNA sequence is that you're trying to amplify. The reverse primer needs to be designed so that it's complementary and the reverse of the coding strand sequence. I believe there are exercises for you to do primer design later in the module, so I'm not going to get into too much detail about how you do that. Again, it's the sort of thing you can look at in the wider reading. It's not as complex as it sounds there, and we'll maybe go through it in a little bit more detail in this lecture as well later on, if we've got time. The other thing to realise is that ideally primers should have similar annealing temperatures. So remember, the second stage of the PCR is to drop the temperature to a point where the primers will anneal to the template. So you don't want a primer in the forward orientation that will anneal at 60 degrees and one in the reverse orientation that will anneal at 50 degrees. Because in order to get the reaction to work, then you have to drop the temperature to the lowest common denominator for the annealing step, i.e. 50 degrees. And that can mean that the primer that would anneal at 60 has a potential now to anneal non-specifically to other regions, because you're reducing the actual stringency of the reaction with respect to the annealing temperature of the forward primer. So just to give you a little example then, this would be a forward primer here. This is a region of DNA, 5' to 3'. So if we were designing a reverse primer to this region of DNA, you wouldn't design it as AGGCTT like you would if you were designing the forward primer. You would have to do the complement and the reverse of the sequence you're interested in. So in other words, you have to do the reverse, so we'd start at this end, and the primer would need to be the complementary sequence. So it will be T, T, G, G, A, A, just like we see here. T, T, G, G, A, A, and then C, A, A, G, C, C, T, C, A, A, G, C, C, T. Yeah? So it's fairly easy once you've gone through a couple of examples to get what you need to do to design these primers, and if you draw the things out on a double-stranded piece of DNA sequence, you'll see why you need to do it like that, because by designing it in this orientation, it will cause the primer to anneal in such a way that it will copy the non-coding strand in a 5' to 3' direction. So the polymerase enzyme itself is obviously very important. We've already talked about its thermostable properties. This is the enzyme responsible for actually copying the DNA, for making new DNA from the template. It's usually Taq polymerase. This is the most common. It's very cheap. It works well. However, in certain circumstances, other heat-stable polymerases are used. It's sometimes important that the polymerase you're using has a very high fidelity. In other words, it doesn't make any mistakes when it copies the DNA. One disadvantage of Taq polymerase is that it does not have a proofreading mechanism. So some polymerases, such as PFU, proofreading polymerases, are able to kind of double check every base that they add during a PCR in order to make sure it's definitely the correct base. In other words, it's definitely the base that's complementary to the template. And it does this by virtue of having a 3' to 5' exonuclease activity. So if it makes a mistake and adds a T opposite a G, for example, instead of adding a C opposite that G, it can remove the mistake and then add the correct base. A PFU polymerase. Taq can't do that. DNTPs, this is the actual nuts and bolts of what gets added to the growing DNA chain. And they come in 4 flavours: deoxy, ATP, GTP, CTP and TTP. And finally, the reaction is made up to a volume with water. Water's there because it's the medium for the reaction. We know that water is effectively what makes up most living organisms. Larger volumes of water can actually help with the reaction, because again it dilutes away impurities that are present. So if you've got a very small reaction volume, it can concentrate those impurities, simply dilute that into a larger volume of reaction. Those impurities get diluted away, and that can help getting the PCR to work. So a little bit more about primer design then. I guess I've kind of covered this a little bit already. I've talked over these points a little bit, but I'll go over them again because I think it's a confusing aspect of PCR and it won't do any harm in this podcast to cover it twice. Effectively, you need a specific forward and reverse primer. The reverse primer must prime 3' to 5', like we've said, and therefore it needs to be the reverse and complementary sequence of the target. These things should have comparable annealing temperatures, and in reality we don't do this thing by eye and just pluck these sequences out of thin air. You can use algorithms and online software now to help in primer design, and there's links to a couple of examples of that here. So just to talk through this actual example what's going on up here, very simple example for designing primers. We've got a gene here. This is the ATG of the gene, and this is the termination codon. Obviously the gene is found on a double-stranded piece of DNA. The coding strand encodes the gene sequence in a 5' to 3' manner. The non-coding strand runs 3' to 5' against the coding strand. Yeah. So the 5' primer design that we will design here will just be as the DNA sequence as it appears in the coding strand of the template. So you would never have a primer, obviously, that is 3 bases long, because it wouldn't work. Generally, they need to be at least 18, so that they're specific enough to only recognise one thing in the human genome. But for this example, I just want to keep it simple. So we've designed a primer that recognises just 3 bases in the gene here, namely the ATG. And then we've designed a reverse primer, and we want that to basically prime from the very end of the gene, this TAA. But we don't design the sequence of that reverse primer as TAA, we need to do the reverse in the complement, so it's TTA for that sequence. Now what happens when you melt these 2 strands and anneal those primers is the 5' primer, which has got an ATG sequence, will recognise the complementary sequence that exists in the 3' melted strand of the template here. So opposite this A there will be a T, opposite this T there will be an A, and opposite this G there will be a C. And the 5' primer will bind to that complementary T, A and C region to prime off the non-coding strand here, and when the PCR proceeds it will copy a replicate of this strand thus. Now the TTA primer we've designed, that will anneal to the end of the gene in the coding strand. So the primer will flip over, TTA will bind to AAT, and then remember that we've got the 5' end of this reverse primer will be here, and the 3' end of the reverse primer will be here, much like it is in the non-coding strand down here, which normally anneals to this region. So what that means is, because it's annealed in this fashion, 5' to 3', we can carry out PCR in this direction. So remember, DNA replication only ever occurs 5' to 3'. So because it's annealed in this orientation, we can go from 5' to 3' and carry out PCR here. So what you have during the PCR reaction is you have the forward primer binding 5' to 3' against the non-coding strand and getting copied in this direction, and then the reverse primer binding to the coding strand, 5' to 3', and going in this direction. So every cycle of the PCR, you double the number of templates that you began with. So if you begin with 2, at the end you've got 4, the next cycle you'll have 8, so on and so forth. What this is down here is a simple formula for calculating the melting temperature of oligos. You can have a look at that in your leisure. But the reality is, again, you get informed of the melting temperature of any primers you design these days from the companies you buy them from, or there are excellent online software programs which you can use for free to determine the actual melting temperature of your primers based on their sequence. So what does PCR look like? Well, the easiest way of seeing whether your PCR reactions works once you've run the cycle in a machine is to run it on a gel. So you're looking to see if the PCR initially has amplified any DNA, and then importantly, has it amplified the DNA that you want? Is the DNA correct? So because we designed the primers and we know where those primers are going to bind in the genome, we already know the predicted size of the PCR products. We know how big a region of DNA we're trying to copy and amplify. So say, for example, the gene you're trying to PCR is 1,000 base pairs. The PCR product we're looking for on an Agarose gel will be 1,000 base pairs in size. So if we have something on a gel that's 5,000 base pairs in size, we've PCR'd something, but it's not the right thing. So in order to separate PCR reactions, and in fact DNA in general, we can do this by something called Agarose gel electrophoresis. I'm not going to go into the details of this because you have a practical on it later in the semester, but effectively the DNA is run through a jelly-like substance which is effectively a matrix through which the DNA can move. Smaller pieces of DNA move through the matrix easier than larger pieces of DNA. Thus, the gel separates DNA according to size. That's exemplified nicely here. These are DNA ladder markers. Each fragment represents a piece of DNA of a specific size, which we can use for determining the size then of our PCR products. The reason we can see the PCR products is that the gels are stained with something called ethidium bromide. We'll mention that in the DNA damage lecture actually later in the module. Ethidium bromide intercalates between the base pairs of DNA and fluoresces when hit with UV light when bound to DNA. So the reason we see these bright bands here is because the ethidium bromide is bound to the DNA, and that's causing the DNA to fluoresce when we hit this gel with UV. You'll see all this in the practical later in the semester. So what we're actually looking at here at the top is a PCR that has worked very well. We have a positive control for the PCR here that we've got a lot of PCR product present for, a huge amount of DNA. And then we've tried to amplify the same gene from several samples, and for some of those samples we can see a clear single DNA species PCR product of a specific size, which tells us that in those particular samples that gene is present. Importantly, we also have a negative control here, which is blank. If this negative control gave a band, you would start questioning the data, because it's possible that the samples have been contaminated, perhaps by yourself, skin cells, etc., falling into the tubes during the setting up of the experiment. You can also see here, this is just a company trying to show you how great their particular PCR enzyme is, but it exemplifies a nice point at the bottom that I want to talk about briefly. These are the PCR products. These smaller brands here aren't actually PCR products. There's something called primer dimers, and this is where the PCR doesn't work very well, and the primers give you a band on the gel due to the fact that the primers have annealed to one another. So this is quite a common problem in PCR, and is one of the many things that you need to troubleshoot when actually setting a PCR reaction up for yourself. So speaking of troubleshooting PCRs, how do we actually go about doing that? Well, there are several aspects of a PCR that can be optimized. The first thing that you can try changing is the annealing temperature of the PCR. So I've already mentioned the fact that depending on the sequence of the primers that you use, the primers will anneal to the template DNA at a specific temperature. So if you design a primer pair and they both have the same melting temperature, that temperature is 60 degrees. If after you've separated the template strands by heating them to 94 degrees, you drop the annealing temperature of the reaction in the next step down to 65 degrees, in other words 5 degrees hotter than the annealing temperature of your primers, chances are that the conditions in the tube will not be conducive for those primers to be able to anneal to the actual template. So the temperature is still too hot for the hydrogen bonds to form between the primer base pairs and the complementary base pairs that exist in the target template DNA. If you drop it to 60 degrees, the temperature is just on the cusp of where those hydrogen bonds can form, and that primer can anneal in a complementary fashion to the template DNA. You might think, well, why take a chance, just drop it to 50 degrees? And then you know that the primers are definitely going to anneal. That's right, they will definitely anneal, but by dropping it a long way below the melting temperature of the primer, you're allowing the primer potentially to anneal non-specifically to other sequences which aren't 100% complementary to the sequence of DNA that occurs in that primer. That's an important thing to realise. So one thing that you can alter if your PCR isn't working and you're not getting any product is you can reduce the annealing temperature. Be less stringent about how the primer binds to the template. If you're seeing too many non-specific products on your gel, such as we're seeing down here, where there's just a smear of all different size products, one thing that you can do is raise the annealing temperature of this PCR reaction, so you make it more stringent. So the primers really do have to find the sequence which is 100% the same, complementary as those that occur in the primer in order to anneal. The second thing you can alter is the magnesium concentration of the reaction. So we've already talked about how magnesium is important because it's a cofactor for the enzyme, but if you have too much there, it can lead to non-specific products being produced during the PCR, and that's exemplified nicely in this gel here. Here we've got a PCR with one nice specific band for the PCR products we're expecting, and this has got a reaction concentration of 1 millimolar magnesium chloride. If we raise that by just 1 millimolar to 2 millimolar, you can see here that there's all sorts of non-specific products in addition to the correct band, which is still there, being produced in the PCR, and that's not what we want. We don't want a big mishmash of all sorts of things in the PCR reaction at the end of the experiment. We just want one specific species of DNA that we're trying to amplify. So adjusting magnesium concentration can help with optimization of PCR. If you're not seeing anything, increase it. See if you start seeing a product. If you're seeing too many non-specific bands, decrease the magnesium concentration. See if it cleans the PCR up. Another important thing is the amount of template that you use in the PCR reaction. We've already talked about that. Too much can inhibit the PCR if there are impurities present. Obviously you need a certain amount of starting material, but it's very little. And the final thing is enhancing reagents, which are often included in PCR, such as DMSO, dimethyl sulfoxide. These reagents assist in the actual melting of the template DNA. So we know we can melt DNA by heating it to 94 degrees. However, sometimes that process isn't 100% efficient, and the inclusion of compounds such as DMSO to the reaction help the template, double-stranded template DNA melt in the first place, which means you get better priming, more efficient PCR. So that's just some of the ways you can modify a PCR. You can look in the textbooks and core text and online to find other aspects of how you can troubleshoot PCRs. They're often excellent examples of how you troubleshoot PCRs from the companies that sell these PCR enzymes. So again, like I mentioned in the lecture on epitope tags, etc., often the best source material for extra reading is a company websites that actually produce and sell these enzymes to researchers. So then just to finish with just some words from another guy about just how important PCR is to molecular biology. Really, to put it into context, try and understand a world, or try and imagine a world, without copy machines or printers, where every paper, report and book had to be typed out or printed by hand. In biology, the equivalent was researchers and companies having to work with minuscule quantities of DNA and proteins naturally produced by plants, animals and people. So before PCR, we had no way of making more DNA other than to isolate it from the actual source, from the cells, from the plants, from the animals, from the bacteria, whatever. There were no way of artificially, reproducibly creating more DNA to work with. And it's only Mullis's invention, PCR, that's really allowed us to do that in the last 20, 30 years now, I guess. And without it, there would be no modern molecular biology. There would be no Human Genome Project. There would be no molecular genetics module. So you might say that, that would be a good thing. But it's a really, really fundamentally important piece of biology, and it's PCR. So it's very important to understand how it works. So the summary of it is it can make milligram amounts of DNA from picograms of starting material. The clever bit is use of this thermostable DNA polymerase, which allows you to cycle the PCR to do rounds of this denaturation, annealing, extension, denaturation, annealing, extension over and over without compromising the activity of the DNA polymerase. And it's a cornerstone of modern molecular genetics. So that's conventional PCR. Some of it we covered at level 1, so there's some repetition there. There's some new stuff about the reagents, but now I want to talk about something that's a little bit clever and more advanced that came on the scene around 10 years ago. I say clever, but really, for the most part, real-time PCR, or quantitative PCR as it's sometimes called, is in effect just like conventional PCR. It uses the same effective chemistry, the same technology as conventional PCR does. The only really clever bit is how we detect the PCR products at the end of the experiment, or importantly throughout the experiment actually for real-time PCR. So when you do a conventional PCR, one of the things that's a bit of a pain is that you only know if it's worked at the end of the actual experiment when you get the PCR reaction, and as we saw on a couple of slides ago, you load that PCR reaction onto a gel, an Agarose gel, run it, stain it with ethylene bromide and see whether you have a band of the correct size. In real-time PCR, the process is very different. We detect whether or not we're getting the correct PCR products during the cycle itself, so during the PCR cycle itself. And this affords us exquisite sensitivity and the possibility to produce what's called quantitative data. And I'll talk about how we can use that production of quantitative data to determine levels of gene expression later in a few slides' time. Another technique which goes hand in hand with real-time PCR is this reverse transcriptase process, and this is a mechanism by which we're able to convert mRNA, which we can isolate from the cell, into a cDNA, complementary DNA copy, which can then be used for PCR. One thing I do not want to see anybody write anything ever in an exam question is PCRing from mRNA or from. I've seen people write PCRing from proteins, which people who write that are beyond help, to be honest. But you must understand you can only PCR from a DNA template. It's the only way it works. So if you're interested in trying to use PCR to detect mRNA levels, you must first convert that mRNA into cDNA, and that's done via this reverse transcriptase process, this reverse transcriptase enzyme. So how do we actually reverse transcribe the mRNA then? Well, we use, as I've just alluded to, this reverse transcriptase enzyme. It was originally identified in viruses, HIV virus, retroviruses. These are viruses which have RNA genomes. In order to replicate their genomes, copy more genomes, they first have to convert that RNA genome into DNA. The DNA is then replicated and made back into an RNA copy. And the enzyme which does that is reverse transcriptase. So what reverse transcriptase does is it takes the mRNA, uses it as a template, and copies cDNA from that mRNA template. So when you use reverse transcriptase in an experimental situation in the lab, you're effectively creating a DNA copy of an mRNA template using the reverse transcriptase enzyme, which is a kind of a clever type of DNA polymerase. There are different ways of priming from mRNA. You should all be aware again by now that mRNAs in mammalian cells and human cells possess this polyadenylated tail. We mentioned it briefly again in the previous lecture on eukaryotic expression vectors. So one way that you can prime from an mRNA is to use a primer which is called an oligo DT primer. It's effectively a run of T residues, and those T residues bind to the polyadenylated tails, which is found in all mRNA, and acts as a primer template then for the reverse transcriptase enzyme to come along and copy a cDNA from that mRNA molecule. There are other examples like random hexamer primers, gene-specific primers you can read about in the Cortex and online, some of the wider reading that's available. So let's say we've generated a cDNA copy of some mRNA then, and we want to use that cDNA now as a template in a conventional PCR reaction. So this was used for numerous applications, such as constructing so-called cDNA libraries for gene expression profiling, for the cloning and expression of eukaryotic genes in prokaryotic cells, which we talked about briefly in the last lecture. But the limitations of conventional PCR remain. In other words, we can only detect whether the PCR's worked, whether we've managed to amplify something from these cDNA copies at the end of the actual reaction, at the end of the experiment. So in other words, there's post-processing required of the sample. The other thing to realise is that when you look on a gel, a DNA band from a PCR reaction, you're actually looking at what's been detected with the so-called plateau range of the experiment. So if you think about what's going on during a PCR reaction, you've got this exponential increase in the number of DNA molecules present in the tube. So every time you do a cycle in the PCR, it doubles the number of DNA molecules present if it's working properly. Now that's true to a point. However, eventually what happens in the actual PCR reaction is the number of DNA molecules begins to plateau. It plateaus for several reasons, including the fact that some of the reagents may become limiting, the DNTPs, the activity of the enzyme, etc. But the important thing to realise is that when you look at what a PCR product's on a gel at the end of a conventional PCR, you're actually detecting the PCR products at the end of the cycle in this plateau range here. And you can get some variability in the amount of PCR product that you get, but generally not much. And the variability that is there is difficult to quantify accurately. So real-time PCR differs massively in the respect that rather than detecting whether or not you have a PCR product at the end of the reaction, and when looking at the plateau range, you actually detect whether or not you're getting PCR products throughout the reaction. So you're detecting during the exponential range of the PCR. And there are two major techniques and technologies which exist that I want to tell you about a little bit about today that allow you to do this real-time detection of PCR products. The first is SYBR Green dye and the second is TaqMan probes. The two work via very different methods. However, the major advantage of each is that SYBR Green is cheap and sensitive, and the major advantage of the TaqMan probe is it's very specific, and it gives you the ability to do something called multiplex in the PCRs, which is to do PCR against several different targets in the same tube. So how do they work? Well, in essence, all SYBR Green is a dye that binds to the minor groove of double-stranded DNA. And when it binds to the double-stranded groove of DNA, it will fluoresce. So it's similar to ethidium bromide in that respect. So what happens in a real-time PCR is that as the PCR progresses, SYBR Green binds to newly amplified PCR products. And if you shine blue light onto the PCR reaction after each cycle, any SYBR Green bound to these PCR products will fluoresce and give off a certain amount of light. So in other words, the amount of light given off by the SYBR Green is directly proportional to the amount of double-stranded DNA in that tube. So if you have a PCR reaction that's working really well, the amount of DNA is doubling every cycle. Every cycle when you shoot blue light at that PCR reaction, you should see an ever increasing amount of this green light coming off from the SYBR Green molecules. Now there's a big limitation to SYBR Green, and that's because SYBR Green binds to any double-stranded DNA molecule, including false positive signals which can be produced from non-specific PCR bands. So whilst you might be seeing a green signal from your reaction, you're not sure whether you're seeing that because your actual PCR product is being produced, or whether or not you're actually detecting the production of these non-specific PCR species, which we saw on the gel earlier on when we were talking about troubleshooting PCR reactions. So TaqMan probes are more complex. Another example is another type of technology called molecular beacons. As you can tell from the registered trademark, no doubt TaqMan is a commercialised name for these, so you can look online again at the company that sell these things, and there's information on there for wider reading. I'm not going to go into a massive amount of detail about how this works, other than to say it uses a technology called FRET, Fluorescence Resonance Energy Transfer. So that sounds horrendously complex, but let me just explain, and hopefully you'll see that it's not that difficult to understand. So how these TaqMan probes work is that they effectively are like a third primer. So you design a TaqMan probe so in such a way that it binds in the middle of the region of DNA which you're interested in amplifying, so between the 2 points where your forward and reverse primer will anneal. So the TaqMan probe will bind in between your forward and reverse primer, and it contains at its 5' end a fluorescent fluorophore, and at the 3' end what's called its quencher molecule. And while it's bound to the template here, the fluorophore will fluoresce and give off light when it's excited with a particular wavelength of light in the machine. But whilst it's bound to the template here, that signal that's given off by the fluorophore will immediately be quenched by the quencher. So in other words, you don't detect any fluorescent signal. However, as your PCR progresses, an extension occurs. So this is the DNA polymerase moving along the template, copying the template DNA. When it encounters this TaqMan probe, the DNA polymerase, by virtue of 5' to 3' exonuclease activity, dislodges and chews up this TaqMan probe, causing the fluorescent part of the probe to be dissociated from the quencher. And when this happens, when you excite the reaction tube with blue light, the fluorescent part of the probe, which is now dissociated from the quencher, is able to emit light, and this can be detected in the machine as a signal. So the beauty of this system is that you will only detect light when this probe is dislodged from the template. So if you detect light, one, you know that the PCR is working because you're producing an extra copy of DNA, and you know that the DNA that you're producing must be the right thing because the TaqMan probe will only bind to the specific gene that you're interested in amplifying. Yeah. So the fluorescence that you generate is directly relevant to the amount of the actual gene target that you're trying to PCR up. This GIF image here exemplifies what actually goes on during a reaction and summarises some of the points I've just talked about. So you can see the TaqMan probe bound to the template here. The PCR comes along, dislodges a reporter which starts emitting light, and that can be detected in the machine. So we've talked briefly about the advantages. These are summarised in a table here. Big advantages of TaqMan are very specific, can be multiplexed, so you can run several of these things in the same tube by simply giving that fluorophore on the 5' end a different colour. So we talked about these different fluorescent tags a little bit in the last lecture on eukaryotic plasmids. Same principle here. Give one TaqMan probe against, say, gene A, a red signal, the other TaqMan probe against gene B, a green one, and the third TaqMan probe against gene C, a yellow one, and you can detect which colour you're seeing after each cycle of the PCR reaction, and depending on how much red, green and yellow, or blue and yellow, whatever, gets emitted from the tube, that gives you an indication about how much of each of those genes is being PCR'd. Not surprisingly, the big disadvantage of TaqMan is the costs associated with designing the probes. SYBR Green is cheap and nasty. It works, it's very sensitive. There are issues with non-specific binding to DNA and therefore false positive results. So, this is a real-time PCR machine. It looks like Geiger's Alien or something, obviously designed by a man. It's quite phallic. This is one of the best ones around at the moment. It costs around 35 grand. So these things are expensive. They're only found really in well-funded research laboratories and obviously companies, pharmaceutical companies, and diagnostic labs in the NHS. For the same price, you could have an RS4. I know which I'd rather have. Don't know about you. So this is a slightly more impressive machine in my opinion. But anyway, I'm digressing slightly there. The point is the Corbett machine and other real-time PCR machines produce data such as what we're seeing here in the right-hand figure. So what you're looking at in this figure is a real-time PCR which is progressing along the bottom here. This is the number of cycles which a PCR is being run at. You have a negative control here, which is purple, I believe. And then you've got different amounts of starting template DNA which has been put into the PCR reaction. So the blue line just had 0.5 nanograms put in, the pink line 5 nanograms, so on and so forth. Now if you look carefully here, you'll see that after about 22 cycles of the PCR reaction which had the most template DNA in starts to fluoresce or give off a fluorescent signal that's detected above this so-called threshold range here. So in other words, it takes fewer cycles to begin to detect a PCR product when we used 500 nanograms of starting material compared to when we used, say, 5 nanograms. Yeah? Now that's pretty straightforward logic. If you've more starting material there, that's the region that you're likely to see a PCR product a little earlier than if you've started with much less material. This actual experiment's derived from tumour samples which have been subjected to real-time PCR, and it's just an example of how little tumour DNA can be detected using this technology. So the detection of residual disease in cancer is very important. This ability to be able to detect a needle in a haystack, one errant tumour cell which has escaped resection via surgery and might be still floating around in the bloodstream. I'll talk a little bit more about what this threshold is here and how it's used to detect gene expression levels. So I mentioned earlier about the plateau phase. This is where we traditionally would detect PCR products on a gel, and I've talked about in real-time PCR how we're detecting the actual production of PCR products during the exponential phase here. And because it's a straight line, obviously you can draw parallels between the amount that you're detecting and the amount of starting material. It's quantifiable. You can do statistics on this sort of data. This is exemplified here. So we've got a 5-fold dilution series here. So this is the same PCR, but the actual starting material has been diluted 5-fold in each case. Now if we were to look at the end result of this PCR, in other words the PCR products which exist in the plateau phase, very little difference in the actual amount of PCR products that we're getting at the end of the PCR here, independent of the actual dilution series that have been carried out. So if you were to run each of these different PCR reactions on a gel, you would see bands for each PCR reaction, and those bands would be of a similar intensity. So you wouldn't really be able to say whether or not you thought that there was any difference in the amount of starting material there in that experiment. However, if we look during the exponential phase, you can see that the PCR reaction with the most starting material again gives us a product far earlier during the cycles of the PCR than ones which have very little starting material. So while during the exponential phase we can see big differences and consistent spacing, if you notice, between each 5-fold dilution, in the plateau phase you can't distinguish between the samples in that way. So it's a nice example of how important it is to actually detect production of PCR products during the exponential phase if you want to actually be able to do quantifiable things with the data. So we've already talked about this threshold, this line which exists above which we tend to start looking at the real-time PCR data, and that's got a specific name. It's called the cycle threshold or CT value. And we use this to assess changes in gene expression prior to and after treatment with drugs, for example. However, in order to do that, in order to really look at gene expression levels, you also need to have something called a reference gene present. So in other words, if you're going to say that the addition of a drug to a particular group of cells caused the expression of a certain gene to increase, you need to have a baseline from which you can make that statement. You need to have a reference gene which doesn't change when you treat with those drugs, so that you can say, look, the baseline, the reference gene remains the same irrespective of whether we treat with the drug or not. However, when we treat with the drug, our gene of interest is expressed at 3-fold higher level. It's actually best to use at least 2 reference genes so that you can have a belt and braces approach and show that neither are affected by the treatment, and it's only the gene of interest that is actually affected in the experiment. So this is shown here, and there's also a link here to some of the more complex nuances around the mathematics for calculating gene expression differences. I don't want to go into too much detail with this. Don't get freaked out by this slide, please. I don't expect you to fully understand the mathematics behind delta CT value, etc. What I want you to appreciate from this is that in order to state that there is a difference between a treated cell and an untreated cell with regards to the expression level of a certain gene, you need to first show that the expression of a reference gene, a housekeeping gene, is not altered in that experiment. Yeah, and that's exemplified here. So the blue and the purple lines represent the reference genes prior to and after treatment with this particular vitamin. You can see the reference gene and in the control and the vitamin treated is very similar with regards to when it goes over the thresholds, when we start seeing PCR products. However, our gene of interest prior to treatment is expressed at a very low level. In other words, only very late in the PCR do we start to detect it. When we treat with the actual vitamin here, suddenly that gene expression is greatly increased because we're able to detect the gene far earlier in the PCR reaction. Yeah. So hopefully you understand that. If you understand that, that's fine. If you want to look into the mathematics of delta CT, feel free, but don't feel as though you need to really fully understand that. Certainly you need to know that it's an appropriate means for testing differences in gene expression. I would consider the knowledge of how it works to come under the remit of extra reading. So how do we use real-time PCR then? Well, it can mostly be used, to be honest, whenever you use traditional PCR. So with the exception really of cloning genes, you wouldn't use real-time to actually bulk up a particular gene for then cloning into an expression construct. It's certainly used now in a whole range of different diagnostic capacities, primarily because of its speed and sensitivity. So you can use it to quantify the amount of virus that are present. So if somebody's going to be infected with a virus, you can do viral load, important for HIV. We just talked about quantifying gene expression levels. So this kind of data can be very important during clinical trials on new chemo drugs, for example. DNA damage measurements can be used. Quality control, pathogen detection, different bacteria can be detected using PCR-based techniques, genotyping. So we can do all sorts of different things with the real-time PCR. And again, you can read about these in the core textbooks and do a little bit more wider reading on some of the applications that exist. One thing I do want to talk about in a bit more detail is this concept of using real-time PCR to detect minimal residual disease. So somewhere where it's particularly useful is something like CML, where we know that the cancer is caused by a particular translocation event to produce a Philadelphia chromosome. So the BCR-ABL chimeric protein here that's produced is unique to tumour cells. So normal cells don't have this fusion protein of BCR-ABL. So if you design primers which span this translocation event and will only PCR a product if this translocation event is present, you can use those primers to detect whether or not you've got any CML cells left following treatment with chemo. A real-time PCR can be used to, in theory, detect one leukemic cell in the body, because a single CML cell is potentially fatal to the individual. If just one cell escapes treatment, that can obviously then re-replicate, amplify up, and lead to remission, lead to a relapse for the patient back to a leukemic state. So I've just summarised some of the advantages here for real-time PCR. I'm not going to go over those again. Again, you can look at these in the actual proper PowerPoint slides if you wish, which are available on Blackboard. So I want to finish on by just quickly going over some hybridization techniques that are also very important for molecular biology. I don't want to spend too long on these because a lot of them now are defunct, but it's important you know about each of these and what they are. So they all tend to work by immobilizing something on a membrane. That membrane is normally nitrocellulose, and that then facilitates a continued analysis of a sample via the specific hybridization of a probe. And the probe is what detects the actual reagent that you're interested in looking at, be that DNA, RNA or protein. And depending on what type of blot you're doing, whether it's southern, northern or western, that probe is different. I'm going to talk about those things briefly in the next few slides. So southern blotting is concerned with the detection of specific DNA sequences, and it was pioneered by Professor Ed Southern. In fact, all the other blotting techniques are kind of named in reference to him. So there wasn't a Johnny Weston and a Frank Northern or a Mohammed Northern who invented those techniques. The reason they're called Western and Northern blotting are because Ed Southern developed southern blotting first. PCR has kind of eclipsed southern blotting now, but it still does offer one major advantage, namely that it can tell you how many times a particular genomic sequence is occurring. PCR will tell you whether that sequence is there in a sample. Southern blotting can often tell you how many times it appears. So if you've got duplication of a gene, for example, in a genome, PCR will tell you that gene's present, might not tell you that it's duplicated. Southern blotting could tell you that. And it works by detecting different sequences of DNA by virtue of a labeled DNA or RNA probe. So what we've got here is basically just an image of how this process works. Southern used an RNA probe. Today we use probes that are PCR derived, so DNA probes. It's important to realise that the probes need to be denatured prior to use. So we're trying to use a probe to anneal to a particular region of DNA that we're interested in. So just like in PCR, the DNA that we're trying to detect needs to be denatured on the membrane so that the DNA becomes single-stranded, and the probe that we're using needs to be denatured so that is single-stranded too. So in effect, when you run the southern blot, you run the DNA out on a gel, separate that DNA on a gel, transfer it to a membrane, and then the membrane is incubated with a labelled probe, single-stranded labelled probe that will recognise a sequence of interest. And that then will be detected via autoradiography. The example shown here is a probe which recognises a specific breakpoint in a chromosome that represents a translocation event. And as you can see here on the gels, control 1 and control 2 don't have that translocation event present. They've only got the one species of DNA as identified in the probe. However, TX15 has got two different DNA species present, and that's because one of the chromosomes in this individual actually contains a translocation event, so it's reduced in size compared to the wild type version. Northern blotting. So this is concerned with the detection of an RNA sequence. Technically very similar to southern blotting. Again, because we can now use real-time PCR to detect mRNA levels, northern blotting is not as important as it once was, but again, some people still consider it the gold standard for looking at RNA because you're actually looking at the RNA itself rather than just inferring how much RNA is there by a PCR technique. You have to separate RNA on a gel that contains formaldehyde. That's because RNA likes to form secondary structures with itself. You know that from just looking at tRNA and the shape that those molecules are. RNA likes to fold up in weird and wonderful ways, so you need to prevent it from doing that, otherwise it can stop the probe which we want to use to detect the RNA from actually working. So you need to run these gels in denaturing conditions, and the denaturing conditions are the inclusion of formaldehyde. We use labelled probes again to detect specific RNAs, so these are probes which have got complementary sequence to the RNA we're interested in and have been labelled with either radioactivity so that they will give a band on an autoradiograph gel, or more recently we've developed non-radioactive techniques which will emit light and also be able to be detected on these autoradiograph films. There's a big problem with RNA degradation. RNA is very difficult to work with. However, with the improved commercial kits that are available now, even project students can extract RNA, as I've just proved in the third and final year projects. Okay, so northern blots then. So northern blots are primarily used, or I guess traditionally primarily used, for detecting changes in gene expression. So from that point of view, they've been largely replaced now, or certainly a lot of research labs have replaced northern blots with quantitative real-time PCR as a way of analysing just how much mRNA is being produced for a particular gene in a cell. However, it's come back to the fore recently as a technique for also analysing the expression of microRNAs, which are a specific type of regulatory RNA that we'll be discussing later on in the module. It's a difficult technique, northern blotting, partly because of how easy it is for RNA to actually get degraded. DNA is an incredibly stable molecule. It takes a lot to degrade DNA. RNA is very unstable. It's very easy for RNA to get degraded and lost during an experimental procedure. So for that reason alone, northern blotting is technically quite tricky. This is a little bit of data here showing a northern blot that was carried out looking at expression of some viral mRNA. Here you've got loading controls. This is another control here. This is against ribosomal RNA. It's one of the RNAs that form the ribosomal machinery. And this is a particular mRNA expressed by a virus. And we can see here different expression levels of the mRNA present depending on whether or not we look at nuclear material or cytoplasmic material. So this is looking at the expression of a particular virus mRNA and whether it resides in the nucleus of a cell or the cytoplasm of a cell. And we can say here in this example that most of the mRNA is in the nucleus and not in the cytoplasm because we have this strong control again, this loading control for the experiment, which shows us that there isn't a lack of viral mRNA here because the experiment's gone wrong, because the loading control is showing a nice level of ribosomal RNA for this sample. So that's similar to the control we had for the RT-PCR on a previous slide. The final technique is Western blotting. This is a technique that is still largely used for detecting protein expression. It's possible to detect the expression of proteins via mass spectrometry. However, Western blotting is still used to confirm experiments which have been obtained using mass spec. So this is still very much at the fore of both research and diagnostics. It works by separating protein samples on a slightly different type of gel to what we separate DNA on, namely this polyacrylamide gel. So polyacrylamide gels are a lot thinner. They're also chemically set. They're slightly different to the matrix which exists for DNA agarose gels. However, the principle's the same. It is effectively a matrix which the proteins are running through, and they tend to run through this matrix according to size. Once you've separated proteins out on a gel according to size, you simply transfer those proteins then across to a membrane, and then you can use antibodies this time as a way of probing the membrane for specific proteins. So remember that antibodies can be raised to be specific against any protein or epitope that you wish, and they're generally specific down to just a single amino acid change in some cases, so it's possible to raise very specific antibodies to even particular mutants of a gene and thus a protein. Detection ultimately generally via this process called chemiluminescence. In essence, all this means is that a secondary antibody is used which recognises the first antibody, and attached to this secondary antibody is a particular chemical group which allows light to be given off when you treat with the chemiluminescent reagent such as luminol. So this is a little bit more information about how you actually go about running a Western blot. Effectively you isolate total proteins from the cells. That total protein is separated via size on this polyacrylamide gel, and then you transfer the proteins from that gel across to a membrane, essentially by just passing an electric current across the gel and the membrane in what's called a Western blotting tank. So this is an SDS page. The Western blot actually refers to the blotting of the protein onto the membrane itself. So there's another GIF here which kind of shows how you detect that protein on the membrane once it's on there. So we discussed on the last slide how we use a primary antibody that's specific for a particular epitope of the protein. However, to be able to see that primary antibody, we then use a secondary antibody which contains a particular enzyme that allows light to be given off. And when light is given off of that secondary enzyme and we expose the membrane to an autoradiograph film, that light results in the presence of a band. So this is what we see here for this Western blot. We have a control here where we're not expressing the GFP protein, and then we have a lane where we're expressing the GFP protein off of the PEGFP expression construct we talked about in the last lecture, and you can see you have here a clear band representative of the GFP protein running at the correct expected size for that protein on the SDS page. Western blots are used for all sorts of things, looking at protein-protein interactions. From a diagnostic point of view, it's still used to diagnose HIV and mad cow's disease, and it's also got an important role to play in proteomics. So this is a 2D gel. Each of these spots is representative of a different protein from the proteome. What you would do in proteomics often is to either treat or untreat cells, say with a drug, and run parallel 2D gels, and you'll be looking for slight shifts in the position of these spots. So it can give you square eyes looking at these things really, but you're looking for slight differences in the presence or absence of these spots, which would indicate the loss or gain of a protein expression as a result of the treatment with the drug. Okay, so just to summarise what we talked about earlier on, that's a real-time PCR is rapidly replacing conventional PCR, and it uses one of two chemistries, SYBR Green or TaqMan PCR. Importantly, real-time PCR detects the PCR product as the actual PCR is running in the machine, so it detects it during the exponential phase of amplification. Because of that, it's truly quantitative, provided that you have the necessary reference gene controls. And it's widely used in both research and diagnostic labs. And also then, just quickly to summarise these hybridization techniques, remember that southern blotting is related to identifying specific sequences of DNA using a labelled probe. Northern blotting, we're interested in identifying specific sequences of RNA using a labelled probe, and in both cases these probes can be DNA or RNA. Western blotting is different. Here we're identifying a specific protein. We don't use a probe, we use an antibody to identify the protein. So they're the main thing. If you don't remember anything about those hybridization techniques, please try and remember that. So I realise that's a long lecture. The podcast has actually gone on a little longer than the actual lecture will have done. One of the advantages of the podcast is obviously I'm not constrained to 50 minutes. So if it's been a bit rambling in places, I apologise for that, but hopefully it'll be useful as a revision aid again for anybody who wants to revisit this lecture. And as I've said before, hopefully I'll be podcasting all of my lectures on molecular genetics this year, so that you can revisit them at your leisure prior to the exam. Thank you.